Hello everyone, welcome. So this is chapter 8 of class 11 NCERT, Cell, the unit of life. So first, what is a cell? A cell in unicellular organisms are capable of independent existence and performing the essential functions of life. So the cell, it is basically the fundamental structure and functional unit of all living organisms. Anton van Leeuwenhoek first saw and described a live cell. Robert Brown later described the nucleus. The invention of microscope and its improvement leading to the electron microscope revealed the structural details of the cell. Then coming to the cell theory. In 1838, Matthias Claydon, a German botanist, examining a large number of plants, observed that all plants are composed of different kinds of cells which form the tissues of the plants. At about the same time, Theodore Schwann, a British zoologist, studied different types of animal cells and reported that the cells has a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is unique characters of the plant cells. On the basis of this, Schwann proposed the hypothesis that bodies of animals and plants, they are composed of cells. So all living organisms are made of cells. That was given by them. Claydon and Schwann together they formulated the cell theory. Later what happened, Rudolf Virchow came into existence, and Rudolf Virchow said that the cells will arise from the pre-existing cells. So that was given by Rudolf Virchow. Okay, so Rudolf Virchow first explained the cell divided and new cells are formed from the pre-existing cell, which is omnis cellula is cellula. So he modified the hypothesis of Claydon and Schwann to give the new cell theory a final shape actually. So it says all living organisms are composed of cells and products of the cells, and next is all cells arise from pre-existing cells. Now coming to an overview of a cell. So a cell, if we see, when we observe an onion cell or if we observe a human cheek cell under a microscope, so we can see it consists of a cell membrane. So onion cell will have a cell wall, whereas the cheek cells will only have a cell membrane. The inside each cell is a dense membrane bound structure which is called a nucleus. The nucleus contains the chromosomes, which in turn contain the genetic material, which is DNA. Cells have membrane-bound nuclei which are called eukaryotic, whereas cells that lack membrane-bound nuclei, they are the prokaryotes. In both prokaryotes and eukaryotes, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both plants and animals. So various chemical reactions that takes place, so it's in the living state. Besides the nucleus, there are other membrane-bound organelles, which is known as organelles like Endoplasmic Reticulum, Golgi complex, Lysosomes, Mitochondria, microtubules, Vacuoles, etc. So ribosomes, they are non-membrane-bound organelles. They are found in all the cells, both eukaryotic and prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also in the two organelles, which is chloroplast and mitochondria. Animal cells contain another non-membranous bound organelle which is called the centrosome. Cells differ greatly in size and shape and activities. For example, mycoplasms. The smallest cell are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometers. Micrometers is 10 raised to minus 6. The cells, they vary in the shapes. They may be disc-like, polygonal, columnar, or any further shapes. Now coming to the prokaryotic cells. In the prokaryotic cells, what is that? That is basically the bacteria, brood green algae, mycoplasma, or PPLO, which is pleuropneumonia-like organisms. So they are smaller, they multiply more rapidly than the eukaryotic cells. So the bacteria, they are of four basic types. They can be rod-like, which is bacillus, coccus, which is pericle, vibrio, which is comma-shaped, and spirulina, which is spiral-shaped. Now, these organisms of the prokaryotic cell is fundamentally similar, even though the prokaryotes exhibit a wide variety of shapes and functions. Okay. All prokaryotes have a cell wall surrounding the cell membrane except in the mycoplasma. The fluid matrix filling the cell is cytoplasm. There is no well-defined nucleus because there is no membrane-bound organelles. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA, they are called as plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher classes, we will learn how the plasmids and all works. Now coming to cell envelope and its modification. So when most of the prokaryotic cell, particularly the bacterial cell, they have chemically complex cell envelope. The cell envelope, they consist of tightly bound three-layered structure. The outermost is glycocalyx, followed by the cell wall and then plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups based on the differences in the cell envelope and the manner in which they respond to the staining procedures developed by Gram. Like those that take up Gram stains, they are Gram positive, and those which do not, they are Gram negative. Glycocalyx differs in the composition and thickness among different bacteria. It could be loose sheath called slime layer in some, while to others it may be thick tough, which is a capsule. The cell wall determines the shape and the cell provides strong structural support to prevent bacteria from bursting and collapsing. The plasma membrane is selectively permeable in nature and interacts with outside the world. The membrane is similar structurally to that of a eukaryote. A special membranous structure is the mesosome, which is formed by the extension of the plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamella. They help in cell wall formation, DNA replication, and distribution of daughter cells. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions in the cytoplasm called chromatophores which contain the pigment. Bacterial cell may be motile or non-motile. If motile, they have thin filamentous extension which is called the flagella. Bacteria shows a range in number and arrangement of flagella. Bacterial flagellum is composed of three parts. One is the filament, the hook, and the basal body. The filament is the longest portion and the extension from the cell surface to outside. Beside flagella, pili and fimbrium are also surface structures on the bacteria, but they do not play a role in motility. The pili are elongated tubular fibers sprouting after the cell. In some bacteria, they are known to help attach bacteria to rocks in streams and also to host tissues. Next, coming to ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 50 nanometers by 20 nanometers in size and are made up of two subunits, which is 50S and 30S subunit, which when present together form 70S. Okay, prokaryotic ribosomes. The ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of a polysome, they translate the mRNA into proteins. Their inclusion bodies, reserve material in the prokaryotic cells, are stored in the cytoplasm in form of inclusion bodies. These are not bound by any membrane system and they lie free in the cytoplasm. Example, phosphate granules or cyanoficane granules and all those things. Next, coming to eukaryotic cells. So, eukaryotic cells are basically membrane. They have membrane-bound organelles, and this eukaryotic cells, they do have a nucleus with an organized nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. All eukaryotic cells are not identical. Plants and animal cells, they are different in cell wall, plastids, vacuoles, and all those things. Okay, so we have an animal cell and a plant cell. First, coming to the cell membrane. So here the detailed structure of the membrane was studied only after the advent of electron microscope in 1950. Meanwhile, chemical studies on the cell membrane, especially in the human red blood cells, enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipid proteins. The major lipids are phospholipids that are arranged polar heads towards the outer side and hydrophobic tails towards the inner side. This ensures that the non-polar tails of saturated hydrocarbons is protected from aqueous environment. In addition to phospholipid membranes, also contains cholesterol. Later, biochemical investigations clearly revealed that the cell membrane also composed of proteins and carbohydrates. The ratio of protein and carbohydrate varies considerably in different cell types. In human beings, the membrane of erythrocytes has approximately 52% of protein and 40% of lipids. Depending on ease of extraction, membrane proteins can be classified as integral and the peripheral. Peripheral means which are on the surface, and integral are which are totally buried inside the membrane. So if we talk about the cell membrane, it is basically the fluid mosaic model. So according to this, quasi-fluid nature of the lipid enables the lateral movement of the proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the protein of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of plasma membrane is transport of molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, the higher concentration to lower. Water may also move across the membrane from higher to a lower concentration. Movement of water by diffusion, that is known as osmosis. As the polar nucleus, polar molecules cannot pass through the non-polar lipid bilayer, they require carrier proteins of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is lower to higher. Such transport is energy dependent. in which ATP is utilized, and that is why it is called as active transport. Next, let us come to cell wall. So a cell wall, it gives a shape, as we were telling earlier, it gives a shape and it also protects from the mechanical damage and infections. It also helps cell to cell interaction and provides barriers to undesirable molecules. Algae have cell wall, which is made up of cellulose, galactans, mannans, minerals like calcium carbonate, while other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of a young plant cell, the primary wall is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner side of the cell. The middle lamella is the layer mainly of calcium pectate, which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata, which connects the cytoplasm of neighboring cells. Next, we have endomembrane system, the membranes inside. So, herein we have many of the membranes. They are considered together as endomembrane system. The endomembrane system includes endoplasmic reticulum, Golgi, lysosomes, and all those things. The first coming to endoplasmic reticulum. So in endoplasmic reticulum, that also we have identified after the discovery of or after the invention of electron microscope. So this endoplasmic reticulum divides the intercellular space into two distinct compartments, that is luminal, that is inside endoplasmic reticulum, and extraluminal, that is the cytoplasm compartment. The endoplasmic reticulum often shows ribosomes attached to the outer surface. So these endoplasmic reticulum which bears ribosomes on the surface, that is called as rough endoplasmic reticulum, that is RER. And if ribosomes are absent, then it's called smooth endoplasmic reticulum. Rough endoplasmic reticulum is frequently observed in cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membranes of the nucleus. The smooth endoplasmic reticulum is the major site for the synthesis of lipids. In animals, cells, lipid-like steroidal hormones are synthesized in smooth endoplasmic reticulum. Next, coming to Golgi apparatus. So, Camillo Golgi, he observed some densely stained reticular structures near the nucleus. These were later named Golgi bodies after his name, Camillo Golgi. They consist of many flat disc-shaped sacs or cisternae of 0.5 micrometer to 1.0 micrometers. Okay. The Golgi cisternae are concentrically arranged near the nucleus with distant convex cis or forming phase and convex trans or maturing phase. The cis and trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicle from the endoplasmic reticulum fuse with cis phase of the Golgi apparatus and move towards the maturing phase. This explains why Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is important site for the formation of glycoproteins and glycolipids. Next, coming to lysosomes. So these lysosomes, they are formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles, they have been found to be very rich in almost all types of hydrolytic enzymes, hydrolases, lipases, proteases, carbohydrates, all those things. Okay. So these enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Next, coming to the vacuoles. The vacuoles, it is a membrane-bound space which is found in cytoplasm. It contains water, sap, excretory products, and other materials not useful for the cell. The vacuole is bound by single membrane called tonoplast. In plant cell, the vacuoles can occupy 90% of the volume. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradient into the vacuole. Hence, their concentrations is significantly higher in vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. Next, coming to mitochondria. Mitochondria, it is specifically stained, and unless specifically stained, it is not easily visible under microscope. So the number of mitochondria per cell is also variable. So the mitochondria is basically double membrane with outer membrane and inner membrane, and it has got two, dividing its lumen into two aqueous compartment, outer compartment and inner compartment. The inner compartment is filled with dense homogeneous substance which is called as matrix inside the green one. The outer membrane forms continuous limiting boundary of the organelles. The inner membrane forms a number of infoldings which is known as cristae towards the matrix. The cristae increase the surface area. The two membranes have their own specific enzymes associated with mitochondrial functions. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called as the powerhouse of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecules, ribosomes which is 70S, and components required for the synthesis of proteins. The mitochondria divide by fission. Next, we have the plastids. So plastids are found in all plant cells and euglenoids. So it can be easily observed under microscope as well. So basis on the pigment it has, the plastids can be chloroplast, chromoplast, and leucoplast. So the chloroplast, which contains chlorophyll, are carotenoid pigments which are responsible for trapping sunlight for photosynthesis. In chromoplast, fat-soluble carotenoids, pigment like carotenes, xanthophylls, they are present, and this gives part of the plant a yellow, orange, or red color. And then we have the leucoplast, which is various size and all. It's basically for storing of the nutrients. Amyloplast, which store carbohydrates. Elioplast, which stores oils and fats, whereas aleuroplast stores proteins. Majority of the chloroplast of green plant are found in the mesophyll cells of the leaves. These are lens-shaped oval, spherical, discoid, or even ribbon-like organelles having variable length and width. Their number varies from 1 per cell to 1 per cell of the Chlamydomonas, a green algae, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called stroma. A number of organized flattened membranous sacs called thylakoids are present in the stroma. Thylakoids are arranged in stacks like the pile of coins, which is called as grana or intergranal thylakoids. In addition, there are flat membranous tubules called stroma lamella connecting the thylakoids of different grana. These membranes of the thylakoids enclose a space called a lumen. The stroma of the chloroplast contains enzyme required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller than the cytoplasmic ribosomes. Then we have the ribosomes. They are granular structures. First observed under electron microscope as dense particles by George Palade. So they are composed of RNA and proteins and are not surrounded by membrane. The eukaryotic ribosomes are 80S while the prokaryotic is 70S. Each ribosome has two subunits, larger subunit and the smaller subunit. The two subunits for 80S are 60S and 30S. There are 60S and 40S, and whereas 70S is 50S and 30S. Here S is Swedberg's unit, which stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. Next we have cytoskeleton. So an elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments and intermediate filaments present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton in the cell are involved in many functions such as mechanical support, mobility, maintenance of shape of the cell, etc. Next, coming to Celia and Flagella. Cilia and flagella, they are hair-like outgrowths. So cilia is small structures which work like oars that you sail a boat, right? And flagella is comparatively longer that we have seen. The electron microscope study of a cilium and flagellum shows that they are covered with plasma membrane. Their core, called axoneme, possess a number of microtubules running parallel to a long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such arrangement of axonemal microtubules is referred to as 9+2 array. The central tubules are connected by bridge and is also enclosed by central sheath, which is connected to one of the tubules of each peripheral doublets by radial stroke. Thus, there are 9 radial strokes. The peripheral doublet are also interconnected by linkers. Both the cilium and flagellum emerge from the centriole-like structure called basal bodies. Next, we have centrosomes and centrioles. So, centrosome is an organelle usually containing two cylindrical structure called centrioles. They are surrounded by amorphous pericentriolar material. Both the centrioles in a centrosome lie perpendicular to each other, in which each has organization like cartwheel. They are made up of 9 evenly spaced microfibrils of tubulin protein. Each of the peripheral fibril is a triplet that is in groups of 3. The adjacent triplet is also linked. The central part of the proximal region and the centriole is also proteinaceous, which is called, and it is called the hub, which is connected with the tubules of the peripheral triplet by a radial spoke made of proteins. The centrioles form the basal body of the cilia, flagella and spindle fibers and gives rise to spindle apparatus during cell divisions and animal cells. Next we have the nucleus. So nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later the material of the nucleus stained by the basic dye was given the name chromatin by Fleming. The interface nucleus has highly extended and elaborated nucleoprotein, which is called the chromatin, nuclear matrix, and one or more spherical bodies called as nucleoli. Electron microscope, it revealed that the nuclear envelope, which consists of 2 parallel membranes with a space between, is called the perinuclear space, forms a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores which are formed by fusion of 2 membranes. These nuclear pores are passages through which movement of RNA and protein molecule takes place in both directions between the nucleus and the cytoplasm. Normally, there is only 1 nucleus per cell. Variations in the number of nuclei are also frequently observed. Some mature cells even lack nucleus, erythrocyte of many mammals, and sieve tubes of the cells of vascular plants. The nuclear matrix or nucleoplasm, which contains nucleolus and chromatin. The nucleoli are spherical structures and present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not membrane bound structures. It is a site of active ribosomal RNA synthesis. Larger and more numerous nuclei are present in the cell actively carrying out protein synthesis. Here there are chromosomes. So there are different stages of cell division. So there are chromosomes. Chromatin contains DNA, some basic proteins called histones and some non-histone proteins and also RNA. A single human cell has approximately 2 meters long thread of DNA distributed among 46 chromosomes. Every chromosome, they have a primary constriction which is known as centromere, on the side of which disc-shaped structures called kinetochores are present. Centromere holds the 2 chromatids of the chromosome. Based on the position of the centromere, the chromosomes are classified into 4 types. One is metacentric, where it is in the middle, the centromere is in the middle. Next, we have submetacentric, where the centromere is slightly away from the middle of the chromosome, resulting in one shorter arm and one longer arm. And next is acrocentric. The chromosome, here the centromere is situated close to the end, forming one extremely short arm and one very long arm, whereas telocentric, it is at the terminal position. Next, there are sometimes few chromosomes, they have non-staining secondary constrictions at a constant location. This gives the appearance of small fragments called the satellite. So that is why it's known as satellite chromosome. Then we have microbodies, many membranes bound minute vesicles Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plants and animals. So that's it. Thank you.